Hello everyone, it's Dr. Gutierrez. Today I'm going to do the second lecture, which is pretty much the basics of cell. And with this, you'll actually get a lot of the things we've talked about in the previous lecture, which I told you to write down on a list so it would help you get back to what you're looking at. I will draw a lot of the pictures that I'm talking about. Now, there's a quick review that I want you to know. One of these are words that you should know and should have in front of you at all times when you're studying physiology until you know the words perfectly. The first one I tell you is to know osmosis, which is a movement of fluid through a semi-permeable membrane based on the solutes that are dissolved inside. I also told you to remember diffusion, which is particles moving from an area of low concentration to high concentration. There's also something called osmolarity, which is the amount of moles in a liter that are dissolved. So the thing is, if you have sodium chloride, 1 mole of sodium chloride, when it's in there, it actually acts as 1 mole of sodium, 1 mole of chloride, so it have an osmolarity of 2. The fluid mosaic model is something I told you to know. Again, it's pretty much a cell membrane having the phospholipid, the cholesterol, the proteins, and all the other things mixed inside of it. Tonicity is pretty much if you have a semi-permeable membrane or a cell and you have that with another substance, another liquid next to another liquid, what you would have is you would have a tonicity based on how much particles are dissolved. And we talked about how you have hypotonic, which means that one of them would have a lower concentration of things dissolved in it. Hypertonic, it would have more concentration, or isotonic, which would be the same. We talked about solubility, where usually we deal with water soluble or hydrophilic or hydrophobic, and amphipathic, which is both hydrophobic and hydrophilic. We also talked about ions, and you have cations, which are the positive charge ions, and the anions, which are negative charge. I also expect you to understand reduction, which is usually having putting hydrogens on something, oxidation, placing oxygen on something, and dehydration would be you're breaking something by adding water into it. Hydrogen bonds are actually important. They're weak bonds, but they form between two molecules that have hydrogens, and so they pull together. Covalent bonds are extremely important because that's where energy is stored, and pH stands for potential hydrogen. And that's important to look at because a lot of times we are going to deal with things being in a specific pH. Now, to review organic chemistry, there are a lot of molecules that we talked about. Organic chemistry is pretty much the chemistry of carbons. So you have carbons, hydrogens, and oxygens in different ratios. We talked about hydrocarbons before, hydro meaning hydrogen, carbon for the carbons, and so it's just chains of carbons and hydrogens, and usually these are hydrophobic, also called lipophilic. You have fatty acids, which are hydrocarbons with a carboxylic acid group in it. And when we're dealing with a carboxylic acid group, what we're dealing with is something that has a carbon on one side, a oxygen double bonded on one side of this. So if you notice, we have 3 bonds, and we know carbon has 3 bonds, and we have another area here with a oxygen and a hydrogen on it. And as you can lose the hydrogen, this is a acid. Now, we sometimes will talk about things being either saturated or unsaturated. And saturated pretty much means it has all the possible hydrogens that a carbon can bind associated with it. Unsaturated means there's probably a double bond somewhere, which potentially you could add more hydrogens to it. We did talk already about cholesterols, which are lipid rings, carbohydrates, which are water-soluble organic molecules, and I always tell people to look at hexose and ribose. Anytime you see ose on a word, as you can see in hexose, the ose here, or the ribose here, it's going to tell you it's a sugar usually, or some sort of polymer of sugar, which I'll talk about later. Hex means 6, so 6 carbons of sugar. Ribose would be a 5 sugar, and that's actually important for a lot of different things. We talked about amino acids, which are carbons with a carboxylic group. You can see it here. And on the other side, you have the amine group, which usually is a nitrogen with 2 hydrogens associated with it. and it acts as a base. And so the thing is, the acid group of one, the carboxylic acid group and the nitrogen on the other can interact to form, a peptide bond, which is what we actually call the bonds between amino acids that are formed, that will eventually form proteins. Now, another thing to actually remember is mono means one. So monomer is one subunit of a specific organic molecule, and a polymer would be repeating monomers. So in fatty acids, we really don't have any. Fatty acids can be broken down into fatty acids can be broken down a little bit more, but usually when we're dealing with fatty acids, we'll be talking about triglycerides, which are broken down into fatty acids, and what's called a monoglyceride. Polysaccharides, poly means many, saccharide means sugar, so polysaccharides are many sugars, so they're carbohydrates. Proteins are made up of chains of amino acids, so a protein is a polymer of an amino acid. And monomers can be joined together by a lot of times dehydration reactions. We'll talk about that later. When polymer is broken, it's usually a reduction oxidation reaction, meaning you have a reduction adding hydrogens and you are adding oxygen on another side. So what that means is you're actually going to add water to it to get it to break. Now, how is food stuff going to get into your cells? Now, when you think about it, you have to go back to the water compartments in the body. And the one where you add things into is always going to be the plasma. So food stuff has to go into the plasma. But to get into the plasma, it has to go into the pretty much the blood through a way. So the first body compartment that food must enter is the plasma. Now, a lot of these do have cells making up membranes, and so it has to get through the membrane. As these membranes, the phospholipid bilayer, tends to be fats, fatty acids, triglycerides, and cholesterol, do not need a protein channel to help it cross into the body. It does need some way to transport it when it gets to when it's trying to get to the plasma. So you use something called a lacteal, which we'll talk about later. Proteins and polysaccharides are too big to enter this fluid directly, and so they have to be broken down to their simplest components. So polysaccharides will be broken down into simple sugars, monosaccharides, and the proteins will be broken down into amino acids, and then they will be able to get into the plasma. Now, to do that, things have to go in. Now, if you're actually bringing in sugars and proteins, what that would end up doing, and even water will go in this, if these molecules are too big to go through these, it will be really hard for these things to go from plasma to interstitium, especially and so you need some sort of carrier protein. You need it to actually come in here, which is based on the diffusion. And if you cannot get these to be broken down, if they stay in the intestines, that means that you'll actually pull water out of the plasma, causing a dehydration. And what that would do is put more water in the fecal matter, so you'll have a diarrhea. Now, once absorbed, what happens? Well, the fate of fatty acids, lipids, and cholesterol, they enter the intestines via something called the lacteals, which send it directly to the heart. And we'll talk about that a bit more, especially when we get into the digestive system. Amino acids and monosaccharides. Well, when you take anatomy, you talk about how you have the hepatoportovenous system. And the hepatoportovenous system takes the blood from the mesenteric veins and brings it to the liver. So the hydrophilic structures will enter the blood via special transporters, which are protein-based. I will talk more about that later. And they will end up getting to the mesenteric veins, go to the liver, hepatoportal vein, and the liver uses the high concentrations of amino acids and sugars to absorb them. It doesn't really rely on a messenger molecule like everything else. Okay, what happens if the liver cannot absorb the excess sugar? So let's look at that. Let's say there's always some sugar that will not be absorbed by the liver, which enters the bloodstream. This usually is taken care of by releasing insulin, which gets monosaccharides into the cell. In a normal non-diabetic person, will usually have blood levels of about 80 to 90 milligrams per deciliters, but can actually spike as high as 180 milligrams per deciliter immediately after a meal. Based on the fluid compartments, if you actually maintain this 180 milligrams per deciliters, uh, what would why would this be a serious condition? Because if you have more sugars that you can't put into the cell, it means it's going to pull fluid out of the cells, causing a mild amount of dehydration, which will end up causing the fluid to be uh, peed out eventually, and you'll get rid of it. Why are fasting blood sugar levels over 150 deciliters in diabetic worrisome and not necessarily in non-diabetics? Well, in non-diabetics, this 180 is just immediately after the meal. Then it actually goes back down. In diabetics, a lot of times their average can be over 150. Sometimes you can have something with their average being over 200 milligrams per deciliter. Now, everyone should have taken a general bio class where you deal with the cells, but I know that may have been a while, and so it's really good to actually look at what makes up the cell. And if we actually look at it, the basics of the cell membrane is a fluid mosaic model, which I've talked about a few times. You have the phospholipids, you have cholesterol, triglycerides, and proteins. These proteins can be integral or peripheral, meaning they can be part of the cell membrane, the phospholipid bilayer, or they can sit just outside. So if we were going to draw the phospholipids here, what we would do is we could actually draw a nice circle here and just assume that this area here is going to be making the fluid mosaic model, which you'd have the phospholipids, the cholesterol, the triglycerides, and the proteins. Cytoplasm, cyto means usually will mean it means bag. In this case, we're dealing with cell. Plasma is fluid. So cytoplasma is a fluid inside the cell, and it's made up of the cytosol. And you also have the cytoskeleton. Again, cyto means cell. Skeleton is a structure that gives you a protein that gives something that gives you shape. And so cytoskeleton would be a bunch of proteins in this which give you a cell shape. Cytoskeleton would be here. Now, some of the proteins are actually structural. Some of them will allow movement, and they usually will have something called an ATPase associated with it. You'll see this again later. Inside the cytoplasm, we also have intracellular enzymes. So we have proteins in here such as kinases, isomerases, phosphorylases, and you'll notice all of these have ase, which means they're an enzyme. I'll talk about later. You also have ribosomes, and usually when I do ribosomes, I usually draw these little figure eights because they have two subunits, and ribosomes are specialized proteins that are enzymes, and they take RNA and read it into the form of amino acids, so they actually help make proteins inside here. We also have another phospholipid bilayer in here, which holds your genetic material, your DNA. And I'll just draw a little squiggly structure here for our DNA. And so we have our nucleus with the DNA. Nucleus is a phospholipid bilayer, and it does have proteins on it which form pores. And these pores are called nuclear pores. It does have deoxyribose nucleic acid, and one thing I actually do want you to pay attention to is the name. You'll notice ribose here. By having ribose here, it tells you it's a sugar. And the nucleic acid tells you that it has a base, a nucleic acid, which actually is going to vary it a little bit. So this is the inherited generic material. It encodes for all the proteins that are going to find in the cell, and it is copied in a semi-conservative fashion. Semi means partially conservative. What it means is that you end up taking helicase, ACE again is an enzyme, and it takes the double helix of DNA, breaks it, and then allows DNA molecules to come in there. And polymerase, ACE again is an enzyme, polymer makes polymers, so polymerase glues it together. So you have two DNA strands, and a new DNA strand will form with each one of these, giving you two new strands, which actually half of it will have each strand, one of them will have the old strands, each one will have a new strand, and so it's semi-conservative. Now inside there, we also have the endoplasmic reticulum. And the endoplasmic reticulum, I usually do make it from the nucleus, giving you this really long phospholipid bilayer structure here. Always remember, endo, in biology, everything means something. So endo means inside, plasmic means fluid, so it's in the fluid. Reticulum is a mesh or a tubular network. And so the endoplasmic reticulum is a matrix made inside the cell fluid. And you have two types. You have smooth, which is pretty much a phospholipid bilayer, cholesterols and some enzymes, and the rough ones, which will have your ribosomes attached here. Now remember, ribosomes make proteins, so wherever you have rough endoplasmic reticulum, you're making proteins on a phospholipid bilayer. You also do have something called the mitochondria. Now, mitochondria is interesting because it does have its own DNA, and it has two membranes. It has an outer membrane and an inner membrane. Now, the mitochondria makes most of your ATP, and we'll talk about it later. And then we also have another tubular membrane over on the sides called the Golgi apparatus. And what the Golgi apparatus is, it takes vacuoles made by the rough endoplasmic reticulum, little bags of vesicles, and it sends them to the Golgi, and the Golgi can then end up keeping them here and store them. And you may want these things stored here because they may have enzymes that break apart proteins, or they may end up having acid or acid-producing enzymes, or it may actually just be that there are messages that you may want to send out of the cell later. And so that's your basic unit of life, which we'll talk a lot about as we go on. Now, proteins are what are encoded for in your DNA. What usually happens is your DNA is transcribed into RNA. So you have your DNA molecule, and it is copied, and you make a copy of RNA from it, and we call that transcription. The enzyme that helps you do that is a polymer, which is called, and the side one is transcriptase. So remember, transcriptase transcribes. ACE means enzyme. The mRNA leaves the nucleus and enters the cytoplasm. So if you remember the cell, if we have our nucleus here with our genetic material, we might have we'll have a protein pore here. And so the RNA will be able to leave from the nucleus. Make a little DNA thing here. So the mRNA that we make can end up going from the nucleus through the pore to get into the cytoplasm. In the cytoplasm, the RNA molecule that we made from DNA will bind to something called a rRNA, which is a ribosomal RNA, that carries a specific amino acid that can bind to it. And that protein will that will actually go to the ribosomes, which will glue it together. Now, if you need the protein again in a phospholipid bilayer structure, the ribosome that has the RNA and the mRNA and rRNA and amino acid will actually go to the rough endoplasmic reticulum. So it would be a good idea to take our cell membrane, make an expansion here, and so we can have our rough endoplasmic reticulum up here. And so we can actually make proteins either on free ribosomes, just kind of hanging out here, free, and they're just going to stay in the cytoplasm, or on the rough endoplasmic reticulum. So what do these proteins do? There's a lot of things. One thing that I keep on mentioning is that that a lot of them are enzymes. Enzymes, what they do is they speed up reactions. They're catalysts, protein catalysts. They can add structure like the cytoskeleton, and they can also add structure to the things outside the cell. In the interstitium, in the matrix, you can actually use these proteins to give structure. they can form receptors on the cell surface. And so these receptors can end up getting messages to tell the cells to do certain things. They can act like pores, like we found in the nucleus, or they can even be a mixture of pores with enzymes that can break ATP and pump stuff out. So I talked about the matrix, which is also structure, and they can also break ATP to allow movement to occur. Now, where does the energy come from? Well, the easiest way to look at it is to look at the molecules that we break. And I will talk about this later today, how we break glucose for this, but glucose can actually be broken down in the cytoplasm via anaerobic respiration. Remember, AN means without, aerobic means oxygen, but it needs the mitochondria and oxygen to be completely broken down to carbon dioxide and water. Amino acids can actually be broken down for energy, and a lot of times they can be can be deaminated and come in as glucose, or other areas such as it can go into the mitochondria. Fats have to go to the mitochondria to be broken down, and they need oxygen. There are a lot of other different ways you can get temporary energy. So you're going to get energy, and we have the phospholipid bilayer, the cell membrane. Again, lipids make up most of the membrane, but remember it's a mosaic. So lipids, one of the things is it is a phospholipid bilayer. Lipids, fats, can cross a bilayer, but not water-soluble molecules. It can through protein pores, and a lot of these pores will allow water to move in and out uh, just because of the size of water. Protein receptors are another thing that are important. They are used to allow water-soluble molecules to enter the cell via pores sometimes. It can actually bind to certain substrates like receptors and send the message via something called a second messenger due to a conformational change. And some can bind, cause opening for channels, which I mentioned earlier. Now, there are different types of movement that proteins can allow. One of them is one of them that you should know is basic diffusion, and this doesn't allow proteins, doesn't need proteins. This actually happens because things move from high to low concentration. Facilitated diffusion does require proteins, and it's just a protein channel, and it works passively. It pretty much either opens or closes, and if it's open, things move easy. If it's closed, it doesn't allow things to move through. Active transport, when you think of active, it means you need energy. You store the energy in ATP, the bonds within ATP, and so active transport, you break ATP to move things against a concentration. You need energy to move things that don't want to move that way. And then you have something called secondary active transport, which what it is you pair something with something else. So it actually works against the concentration gradient of one thing, but with the concentration gradient of another. The other type is endocytosis and exocytosis. Again, you've noticed that I tend to have you do this. Endo means inside, cyto, cell, and so cytosis is the process. A process of endocytosis would be the process of bringing something into the cell. Exocytosis is a process of taking something out of the cell, and I will talk a lot more about that as we go along. So, again, endocytosis, if a substance can bind to a receptor, it triggers a cytoskeleton to move, pulling the cell membrane in, and so you end up forming a little bag from the cell membrane that gets pulled into the cell, and that's endocytosis. Exocytosis, it's a process of pushing something out of the cell, and this can a lot of times be to send messages that the cell may want to send to something else to make sure that other cells do something to maintain homeostasis. Okay, now I've talked about chemical reactions, but it's really important to actually talk about it. Remember, reactions can be endothermic or exothermic, which means they could release energy or absorb energy. When a bond is broken, it releases energy, so it's an exothermic reaction. When a bond is formed, it takes in energy, net energy, so it's an endothermic reaction. Now, to get any sort of reaction, it doesn't matter if it's a net endothermic or exothermic, you need something called activation energy. And activation energy is the energy you need to get something to start moving in a reaction. Now, when we're dealing with reactions, we're talking about reactants and products. The reactants are what comes in, the products are what goes out. So if we're actually looking at something like glucose with oxygen being used to being broken down for energy, glucose binds to oxygen, and what you end up with is the carbon dioxide and water. So carbon dioxide and water would be the products. Glucose and oxygen, in this case, would be the reactants. Now, I am going to mention this a lot. An enzyme is a catalyst. And a catalyst is something that helps speed up a reaction but is not used in the reaction itself. An enzyme, again, is a protein catalyst. You probably have heard me say that a lot. And what enzymes can do is they can pair reactions together so multiple reactions happen. What they're going to do is, again, they're a protein catalyst, so they decrease the activation energy, which I mentioned earlier. Now, the way they work is enzymes have binding sites for substrates, which actually can vary in a lot of ways. It can be very specific or non-specific. And when the enzyme binds substrates, it actually waits for both of them to come together to do something. And so it compare the reactions. So for instance, when you're making ATP, you have glucose, and this is very simplified. We'll go into it a lot more detailed in a little bit. Can be broken down into pyruvate. As you're breaking this down into pyruvate, you're getting an exothermic reaction. You have ADP, adenosine diphosphate, and you're binding it with inorganic phosphate to get an ATP. As you're forming a bond, it's an endothermic reaction. And so the breaking of the glucose to pyruvate gives you heat or energy, endergonic reaction, and it ends up exergonic reaction, and it ends up actually driving the ADP and the phosphate to form ATP in an endergonic reaction as well. Glucose has to bind to the binding site. ADP and phosphate have to bind to the binding site, and once the enzyme works, they both get released. And so there's actually a lot of the different things that will happen. So if you're breaking glucose to two pyruvates and the ATP will leave when it's done. Now, big thing to look at with enzymes is you'll always see the ACE associated with it. And so if you're breaking ATP, usually you'll see ATPase, which would be a breakdown of ATP. A catalase or peroxidase are enzymes that break down peroxide, and so they catalyze the breakdown of peroxide, and so peroxidase, catalases, different types of enzymes. Helicase is an enzyme that works with the double helix, opening it up. Polymerase, ACE again enzyme, polymer, so it's an enzyme that makes polymers. Kinase is something that moves something. A lot of times the kinases will move phosphates, so we'll deal with those later, but kine means move, ACE is enzyme. Phosphatase is something that works on phosphates. A lipase, ACE is an enzyme. Lip or lip means fat, so lipase is an enzyme that works on fat. Amylase has to deal with the complex carbohydrates. Amylose is actually one of them. And so anytime you see ACE, it's an enzyme that breaks complex carbs. Over here you have proteases. Proteo for protein, ACE is breakdown. Helicokinase is something that's going to is actually going to phosphorylate, move something in a sugar. Ligase is something that an enzyme that glues something. Oxidase will actually work with oxygen. And so just wanted to show you how enzymes were named. Now, there's a couple questions that will help you understand how this works. How do substrates find their binding sites? So what I mean by that is this. You have a protein catalyst with, let's say, two binding sites. One for ATP, that would go in here. ATP, and one for glucose, but G for glucose. So glucose has to find its own binding site, ATP has to find its own binding site, and what allows these to come into their binding site is what allows things to go from an area of high concentration to low, basic diffusion. Now, these have to go into their binding sites, and only by going into their binding sites can they be acted on in the enzyme. So if you have just glucose floating up here, you might break glucose, but it won't be used to make ATP necessarily. Now, can something inhibit an enzyme? The short answer is yes. The long answer is yes, and it can inhibit different ways. One is it can compete for the binding site. So you may have something that looks like glucose that tries to get into the glucose binding site, and in that case, it's going to inhibit glucose from binding in there, making the enzyme not function. You also do have a non-competitive inhibitor, which can bind somewhere else, which causes a change in the protein, which doesn't allow the protein to actually function. Now, why might someone want to inhibit enzyme activity? Well, some of this enzyme activity may be to break down hormones. So you may have something called a um, monoamine oxidase inhibitor, and it breaks down monoamine oxidases, which are neurotransmitters or hormones which can excite the body. And so if you inhibit them, you can actually get the body more excited. You can actually decrease the risk of depression. Or serotonin reuptake inhibitors, selective serotonin reuptake inhibitors. It inhibits an enzyme that brings serotonin back inside the cell, so there's more serotonin to bind to different receptors. The more of more of a messenger molecule that can bind to a receptor, the more likely it is to bind. Now, there are a couple of respirations I want to talk to you about, which I mentioned. One of them is called anaerobic respiration. It's respiration without oxygen. So aerobic means oxygen, with oxygen. Anaerobic would be without. So anaerobic respiration would be respiration without oxygen. What we're dealing with is we're breaking stuff to make energy. Anaerobic respiration would be breaking things down with oxygen. And a lot of times when you're in general bio, we break it down simple. Over here, we'll show you how things are not necessarily as simple as we make them out to be. And so let's deal with basic glycolysis, which is an anaerobic process. Glycolysis, you can take down the word again. Glyco means sugar. Lysis means to break. I would tell you to write the words down so you remember that glyco means sugar, lysis means break. And so in basic glycolysis, you're taking sugar and you're breaking it. There's a lot of steps to it, and I would tell someone, don't necessarily know all the steps right away. We will talk about them more and more, but it will show you how enzymes are used to make energy. Now, at rest, remember the cell membranes, the transmembrane proteins, are actually pumping 3 sodium out, 2 potassium in per each ATP that's broken by an ADP. Remember, this gives you energy. Now, there's more sugar inside the cell than outside usually, but you want to get the sugar in there because that's where it works. So what you have is you have hexoses, and actually even riboses, coming into the cell via protein channels. You remember how I talked about the protein channels in the cell membrane? So what you can have is you can have glucose, which is a hexose, come into the cell with sodium. So you would have an enzyme where glucose will bind, sodium will bind, and insulin would bind, and that would allow the high level of sodium outside of the cell to move in to the cell. And because of that, glucose can follow through, which is it technically is a co-transport, and it's a co-transport which actually uses one concentration of one thing to allow something that's higher concentration to go in. Now, glucose has to be prepared in the cytoplasm, has to be broken down in the cytoplasm and prepared to get it into other areas. And so even before you do something, even before you begin glycolysis, again, glycolysis means breaking sugar, you have to get things ready. This does happen in the cytoplasm, again, the cell fluid outside the nucleus. And I don't necessarily want you to know the name of all the enzymes, but I want you to pay attention to the enzymes just to see how they're called. So glucose has to be phosphorylated, and it's phosphorylated by something called a hexokinase. Hexo for hexokinase, moving ACE, and so it makes a glucose 6 phosphate. And where you get the phosphate from is by breaking one ATP. So we're removing an ATP when we start this. In many ways, you can see this as adding to the activation energy. Then you end up having this glucose 6 phosphate being converted to a fructose 6 phosphate by something called an isomerase. Iso means same, merase. So it's going to change the shape of something, but it's going to keep the same formula. So the fructose will actually still have the 6 carbons. It will still be bound to phosphate. Nothing's going to change. It's just going to change the ring configuration. The fructose 6 phosphate will be phosphorylated by phosphofructokinase. You'll notice kinase is moving a phosphorus onto this usually. And now you have fructose 1,6-bisphosphate, which is going to break, which means you're going to break another ATP. So you are breaking 2 ATPs to start this. And then the fructose bisphosphate is going to be broken down into 2 3-carbon structures, which both of them are going to be converted. There's actually one thing is converted to one thing, but I'm not going to go into that. But they're going to be converted into glyceraldehyde 3-phosphate, which is a glyceraldehyde, glycerol. Aldehyde is actually a functional group as well, with a phosphate on carbon number 3. So this is actually going to be dehydrated by glyceraldehyde 3 dehydrogenase. You should notice, again, dehydrogenase is an enzyme that dehydrogenates. And an coenzyme called nicotinamide adenine dinucleotide, NAD, which usually carries a plus. So this is actually going to dehydrate this, and means that the NAD is going to become an NADH. And that can actually be used to make ATP later, by the way. This dehydrogenate is actually going to the glucose 6 is going to form a 1,3-bisphosphoglycerate, which is going to be dephosphorylated by a phosphoglycerate kinase. Again, you're going to move a phosphorus from the glycerate to form an ATP and a 3-phosphoglycerate. 2 other enzyme reactions occur. I'm not going to talk about those, but what you end up is with a phosphoenol pyruvate, PEP, phosphoenol pyruvate. And the reason I actually like you to see that is because it has a pyruvate. This will be dephosphorylated and give you another ATP and a phosphorus. So you can see you do form this NADH. I'm sorry, this NADH. I was right. It's just not that one. You're going to form 2 ATP gross. So you're going to actually form 4 ATPs at the end, and so you end up getting a net of 2 ATP. And this ATP will help run a lot of those aspects of the cell. Now there is something called a bridge reaction where you don't have any ATP formed, but it's important to move this to the mitochondria. What happens is pyruvate is dehydrated and joined to something called coenzyme A by an enzyme called pyruvate dehydrogenase. Something that dehydrogenates is called the dehydrogenase. And what it forms is a special compound called acetyl-CoA. Now acetyl-CoA is actually very, very important. The other thing that happens is you're going to form 1 carbon dioxide. So pyruvate is a 3-carbon sugar. When it becomes acetyl-CoA, it's a 2-carbon sugar with the CoA, coenzyme A, which does have a lot more carbons. But there's no net energy gained or lost. You just break a carbon off, you glue the coenzyme A onto it, and this occurs in the cytoplasm. And the acetyl-CoA has a couple fates. One is it can go to the mitochondria to be broken down as energy. The other is it can get sent to specialized cells that can make it into fatty acids. And so CoA enzyme A can be used to make fatty acids. Now, if you can make fatty acids with it, you can also take the fatty acids and convert them to acetyl-CoA. So fats can go into the mitochondria and be broken and make more ATP through oxidative phosphorylation, which means it requires, at the end, oxygen. So fats, to be converted into energy, you need to convert them into acetyl-CoA, and you're going to need oxygen to drive that. Now, we do have the citric acid cycle, and you notice I put here that it's anaerobic, meaning you're not going to use oxygen to run this. But a lot of the products are going to need oxygen to end up breaking down to form ATP. So what happens is acetyl-CoA comes into the mitochondria, and it's linked to something called oxacylic acetate by citrate synthase. Again, you have synthase, tells you it synthesizes citrate. And so you end up forming citrate. Citrate is a 6-carbon structure and is converted through to isocitrate by it is actually one of the isomerases, but I actually gave you the exact name, which was aconitase, and that's going to make isocitrate. Now, if you notice, so far you're not getting energy, and this is dehydrated by isocitrate dehydrogenase. Go figure. You could such a enzyme tells you exactly what it does, and when it does that, the hydrogens come off, and they go to the NADH. I'm sorry, the NAD, which ends up taking this, and the isocitrate is converted to alpha-ketoglutarate. I didn't put the alpha here, but the alpha-ketoglutarate is dehydrated, again, by alpha-ketoglutarate dehydrogenase to form another NADH. So the NAD picks up the hydrogens, and succinyl-CoA. Could be succinyl-CoA. The succinyl-CoA has a phosphate removed by succinyl-CoA synthase. This one's actually, the enzyme's working backwards. It can go backwards and forward, apparently. And you end up getting a guanosine diphosphate being formed into a triphosphate. This can actually be converted directly into ATP. And what you end up forming is the succinate. And succinate will then be dehydrated by succinyl dehydrogenase to form fumarate. So what you're seeing is most of the enzymes are actually telling you what they do, roughly. And you end up getting an FADH2. This is a flavin adenine dinucleotide. Flavin is related to riboflavin, by the way. Adenine is the amino acid and the base dinucleate. And so this is actually going to form the FADH2. So it's going to pull 2 off. And so fumarate is going to be hydrated afterwards by fumarase to form malate. Malate then gets dehydrated by malate dehydrogenase to form one last NADH and a oxacylic acetate. And you'll notice that this oxacylic acetate is the same oxacylic acetate that you have over here. And that's why it's called the citric acid cycle. This will actually come up here, and the acetyl-CoA can actually link there to end up forming the citrate. So if you notice, we actually form 1, 2, 3 NADHs, 1 FADH2, and 1 GTP. So all this together is going to actually do things. So far, we've actually have released carbon dioxides. We've actually been releasing carbons, but we haven't actually been using oxygen. The oxygen happens in the internal membrane of the mitochondria, where you take the NADH and FADH2 and you transfer the electrons to oxygen and then form water. And there are a lot of steps in there. There's a lot of coenzymes and a lot of cytosols in there. What I want you to realize right now is the ATP equivalent. So 1 NADH can give you potentially 3 ATP when it goes through the electron transport chain. So you're getting, while the citric acid cycle doesn't make ATP directly, it makes the NADH, which will give you the equivalence. The FADH2 has about 2 equivalent ATPs, and what you're going to get as a byproduct is water at the end. So the oxygen is going to be excited, and the hydrogens from the NADH will move to oxygen. The FADH will also move to the oxygen, and so you're going to get a water. The total amount of energy that you get from one glucose via glycolysis, the citric acid cycle, in the electron transport chain, will be more than 24 ATPs per glucose. There are some other areas you're getting it. There was actually an NADH I didn't mention, which does come in there, and there's a lot of times when you're actually breaking stuff, you get partial, but a lot of time the important thing is when you actually go to the complete breakdown using the glycolysis, citric acid, and all that, you get more than 24 ATPs per glucose molecules. Now, if you think about it, glycolysis, anaerobically, you get 2 ATP net and 2 pyruvate. The 2 pyruvates are going to go to the citric acid cycle, and in the citric acid cycle, you're going to form 3 NADHs. So 3 NADHs as you have 2 pyruvates going in there. And you're going to double that, but 3 NADHs, and for each pyruvate that goes in there, you're going to end up getting 3 too. So you're going to have 3 times 3, so about 9. That's in my math now. NADH, 3 per each pyruvate, so that should be 6, so it'd be 12. I'm sorry, that was right. So 6 times 3 is, yeah, that's 12. That should be 12. FADH, we'll actually get 2 ATP per FAD, so you'll end up having 2 ATP per every pyruvate that goes in here. As you have 2 pyruvates, you'll have 4, and then you'll have the GTP, which just converts to ATP. Now, for that to happen, though, you have to go to the electron transport chain, and I wanted to just give you some questions. Now, what is metabolism that occurs without oxygen? And the answer is anaerobic respiration. What part of the cell does glycolysis occur? Glyco means sugar, lysis means break, and glycolysis happens in the cytoplasm. What is a bridge reaction? It's where you take pyruvate and you convert it to acetyl-CoA so you can bring it into the mitochondria. Where does a citric acid cycle occur? And that is a matrix of the mitochondria. What type of metabolism occurs in the citric acid cycle? It's still anaerobic respiration in the citric acid cycle because you're not using oxygen. What is produced by the citric acid cycle? Well, you have the NADH, an FADH2, and a GTP, and these are going to go to the electron transport chain. Where does the electron transport chain occur? It occurs within the inner membrane of the mitochondria. And does the electron transport chain require oxygen? Yes, it requires a lot of oxygen. And this is a question that should help you think about stuff, and that is, why might decoupling the electron transport chain from the citric acid cycle would be something that would cause death relatively rapidly? And I'm not going to answer that. I'm going to let you answer it, and you can put the answers on the comments below. And I want to put everything together for protein synthesis. So, all proteins normally found in the human body are encoded by nuclear DNA. Remember, the nuclear DNA is transcribed into mRNA by a transcriptase. The mRNA that's made leaves the nucleus via a nuclear pore and goes to the ribosomes, and the ribosomes will translate it into protein. If the protein will eventually leave the cell or can damage the cell, it should be made in a phospholipid bilayer, so it would actually be made in the rough endoplasmic reticulum. The ribosome would move to the rough endoplasmic reticulum. Proteins made in the rough endoplasmic reticulum can be glycosylated. glyco means sugar. So glycosylated means you could add sugar to it. Proteins made in the ER will be sent to the Golgi. Now the Golgi decides where things should go. If this cell needed to be made in the cell in a vacuole for some reason because of how it actually is full, it can be sent to the Golgi, and if this protein was meant to be a protein for the nucleus, a nuclear pore, it will actually be sent to the nuclear envelope for that. Golgi can modify proteins via glycosylation. Again, it's just adding sugar. It's a different type of glycosylation. And the vacuoles of the Golgi can be sent anywhere needed, and they can fuse to the cell membrane. Vacuoles can also be stored as needed. So if we actually draw this again, we have our cell membrane here. We have our nucleus in here. And we have our DNA, our genetic material in here. Our DNA will be copied into RNA and will leave via a nuclear pore, a transmembrane protein here. And so our mRNA will leave. I'll make it purple this time. And so you have your mRNA. The mRNA will bind to a ribosome. And I'll put the ribosome here. And it'll also bind the rough the rRNA, which brings the amino acids over. And the ribosomes will start copying it in something called translation. If this is going to be made in if this is actually is a protein that may actually be harmful to the cell, or if it needs to be made in a phospholipid bilayer for some reason, the ribosomes can go to the endoplasmic reticulum, and so you have rough endoplasmic reticulum forming the proteins. If it's actually these free ones, it can make proteins that are normally found in the body, such as cytoskeleton proteins, giving you structure here. From there, if they're actually made in the endoplasmic reticulum, you can put the proteins in little bags called vacuoles, and the vacuoles can go to another membrane structure called the Golgi, which sorts and packages the cells, and the Golgi can decide what to do with it. It can fuse it with the membrane to form another transmembrane protein in the nuclear envelope. It can actually fuse with the phospholipid bilayer, or it can just keep these proteins inside these vacuoles until needed. I'll put a little vacuole and a little protein in here. Now, if you want to get the protein out, the cytoskeletons can take the vacuoles and move, so the vacuole will end up being moved to the cell membrane, fusing with it and pushing it out, which would be the exocytosis. Now that is no longer protein synthesis, it's how you're bringing stuff in. The cytoskeleton can also act to bring itself in, and if it brings itself in one way and pushes on the other two sides out, what you can end up with is a phospholipid vacuole that comes into the cell, which would be if you're bringing it in, it would be endocytosis. If you're pushing it out, like here, it's exocytosis. And so vacuoles can actually use it to be stored as needed. Now, the breakdown, we talked about how we have the of energy. We talked about how we have mitochondria in here. And mitochondria has an outer membrane. We talked about how glucose came in here via some sort of receptor which binds glucose and sodium to come in here and bringing it together as a co-transport. So glucose will come in here. Glucose will be broken down into a 3-carbon sugar. Well, I'm sorry, it'll be broken down in a 3-carbon structure called pyruvate, and pyruvate will be converted to acetyl-CoA and be moved to the mitochondria, where it's further broken down into carbon dioxide and you form FADH2 and an ADH, which will go into the endomembrane to form your ATPs and also water. And so what you end up getting from a glucose molecule, you end up eventually getting carbon dioxide and water. Now, carbon dioxide is a nonpolar molecule, so it can diffuse across the membrane without a carrier. So as you're breaking down sugar inside the cell and using the mitochondria, you build up carbon dioxide, and through diffusion, carbon dioxide will leave. Now, and you will have some water there too. Now, carbon dioxide has some really neat properties to it. It can actually bind directly with water, forming carbonic acid, and so the acidity inside the cell tends to be a bit lower than acidity outside the cell as well. And so you have cells, you have cells with transmembrane proteins which are pumping things in and out of the cell. We have all these different things, and so at this point you have the basics of the cell. So cell membrane can be pulled stuff in. How does it get fuel? I already talked about. Now, there are a lot of things that you want to remember from this lecture that I would actually tell you to make a sheet, list these, and write them down so you have access to them whenever you need to. Now, one of them is movement in and out of the cell. You have basic diffusion, which would be how carbon dioxide gets in or out of the cell. You have facilitated diffusion, which requires a protein channel. You have active transport, which requires a protein that breaks ATP to move stuff, and so it's an enzyme in essence. You have endocytosis, which I talked about. It's bringing things into the cell by pulling the cytoskeleton in more, pulling the phospholipid bilayer in. Exocytosis, pushing stuff out. It's important to know the formation of proteins. So you remember you have transcription, which is taking the DNA, and through transcriptase, you can end up making a copy of the DNA to mRNA. mRNA will leave, go to the ribosomes, where it'll be translated into protein. And so translation, the ribosomes take the mRNA and copy it in the form of protein. And you have different structures. You have primary, secondary, tertiary, and quaternary, which I've mentioned before. I'll let you look at those. We actually also have chemical reactions that are important. We have endothermic, which means you're going to bring energy into it, usually forming bonds. You have exothermic, which means you're going to break bonds and release energy. You have catalysts, which are things that speed up a reaction by usually decreasing the activation energy. You need activation energy to start any reaction. We have reactants, which are what comes into the chemical reaction, and the product is what we get out of a reaction. Now, in the cell, most of our reactions are catalyzed by enzymes. So enzymes are protein catalysts. What we call reactants in enzymes are usually called substrates, and what you get out is still a product. Now, the substrates have binding sites, and these binding sites are affected by a lot of things. What actually causes things to go in is diffusion, and so heat is actually important. The right pH is important, and the right protein structure is important. So if you destroy the enzyme, you end up having a lot of issues. We'll talk a lot more about enzymes later. I do want you to understand the principles of metabolism. So you have catabolism, which is when you're actually breaking something down for energy, so you get more energy out of it. Anabolism, you're building stuff up, so you're adding energy to this, building it up. Now, I talked about breaking down a glucose, and this is something very important. I would actually tell you to take a blank sheet of paper and go through this where it happens and have it down so you can definitely see it. So you have glucose being broken down via glycolysis, and I went through the entire process. The idea is glucose is going to eventually be broken down into 2 pyruvates and 4 ATP, but you put 2 ATP in. The pyruvate has to be converted into acetyl-CoA, and that's called the bridge reaction. You don't get energy, but you can bring it into the citric acid cycle. And so what you have is you have citrate, and it goes from citrate to isocitrate. From isocitrate, you have an alpha ketoglutarate. Then you have the succinyl-CoA. Then you have the succinate. Then you have the fumarate. Malate. Oxalic acetate. And when a acetyl-CoA comes in here, you end up forming citrate again. Citrate is also citric acid, so that's why it's called the citric acid cycle. Now, I talked about how you are getting some NADH from these reactions. And you are getting carbon dioxide leaving here too. And what ends up happening is the NADH and FADH2 will end up going to the electron transport chain. NADH will give you 3 ATP equivalent, roughly. FADH will give you 2. And a GTP, which actually does come out of here, will actually give you 1 ATP equivalent. And that's actually kind of neat. I actually did this. This was an accident. So if you actually look at NADH, 3, FADH2 gives you 2, and a GTP will give you 1 ATP equivalent. I hope you enjoyed this lecture. I hope you found it usable, and I hope you have a nice day.